Welcome to Under the Microscope. This series is brought to you by the Real Scientists Nano team. Our goal is to provide a platform where scientists can communicate their work and interact with the public. With that in mind, every week we introduce you to a scientist working in the field of materials and nanoscience. Hello everyone. Today we have with us Gregor Fuhrmann, who is the head of a junior research group Biogenic Nanotherapeutics at the Helmholtz Institute for Pharmaceutical Research, Saarland, which belongs to then the Helmholtz Center for Infection Research based in Germany. Hello Gregor, how are you doing?
Hi there, how are you?
Well, dealing with the current situation and excited to talk to you.
That's great.
Shall we begin?
Yes.
Wonderful. So let's start with understanding your scientific journey. So how did you end up in your current research field?
So I did my PhD in Switzerland at ETH Zurich, where I already worked in pharmaceutical sciences. And then after finishing 2013, I moved to London to start as a postdoc there at Imperial College. And there there was another postdoc working in that group who was working on these cell-derived extracellular vesicles, and I became interested in using them for delivery of certain compounds and drugs. And since then, I've been studying them for different applications and different drugs.
That's interesting. So you have had a typical researcher, the traveling researcher, starting in Switzerland, going to UK, now being in Germany. Was it fun though?
Yeah, it was very nice. Yeah. All of the stations were very nice. I met a lot of good friends that I still have right now, and yeah, it was a very nice experience to see these universities and these different ways of how they are approaching things. I think it's very useful.
Yeah, that's wonderful. So, where would you say your current research falls in this big picture of materials or nanoscience, this big puzzle? Where does your research fall?
So my research is also looking into engineering of drug carriers, but we're not following purely synthetic approaches. We are trying to use principles that are already established in nature, biological principles, for example. So basically, we're trying to use up some new methodologies for formulation of, for example, cell-derived nanoparticles. We also are trying to set up some imaging and diagnostic tools to use nanotechnology or to use these extracellular vesicles for detection or of diseases or for therapeutic for treatment opportunities. And our specific focus is on treating infectious diseases, but also autoinflammatory dispositions.
Okay, interesting. So it sounds to me that you do a lot of fun research projects or experiments in the lab. So if you have to pick one, a quirky or the most fun one, or the most you're proud of, I know it's difficult. But if you have to pick one research project, could you pick one and explain it to us in the section we call In Other Words?
Okay, so I think I would pick, there's a very recent paper that we published from one of my PhD students, which I was also very proud of her and of her work. Basically, what we did is we looked into a group of soil-living bacteria that are called myxobacteria. So this is a cooperation with another group in our institute that grow these myxobacteria. And so what we did is we isolated the extracellular vesicles from these myxobacteria and wanted to understand what is their role and can we use them for drug delivery. Now it turned out that if we take these bacteria-derived vesicles, we put them onto mammalian cells, they are really low in toxicity. So even in very high concentrations, they are not killing cells, and in very simple cell models. Although this is bacteria-derived material. And what's even more interesting is that the myxobacteria, they are already shedding or producing the vesicles with an antibiotic compound inside. So we detected an antimicrobial compound, and we showed that these vesicles are, so to say, inherently antimicrobial against certain model pathogens. And that was quite unexpected to see that basically the bacteria are doing our job. They are producing the nanoparticles, and they are also loading them with a new compound or a recently discovered compound. And that's a very nice project that we started, and we're now trying to further characterize these vesicles, also to understand to understand them in, for example, with primary mammalian cells, how they interact with these, and if we can formulate them to really treat infections, for example, in the lung or in the gastrointestinal system.
Wow. So these are the bacteria from the soil, which could in
Yes.
principle be used for as a therapeutic for lung or
Yeah.
whichever.
It's known that these bacteria, they are really potent producers of natural products, and this is what many groups or some groups are looking into. They basically culture the bacteria and they look which kind of drugs are they producing, and there's already drugs that are clinically or that are in clinical development. But it has not really been looked into the vesicles that these bacteria are producing, and it turned out that there's also drugs in the vesicles, and the vesicles are not toxic against cells, at least in our simple tests. So yeah, in principle, we hope that this could be a new nanotechnology avenue to treat bacterial infections.
That's fascinating. That's so cool. That's so I
Thanks.
can imagine, I can understand why you picked this paper to talk about as the one that you're most proud of.
Yes.
That's so interesting. Um, let's speak a little bit of the other sides or other parts of a researcher's life, which
Mm-hmm.
is teaching. So do you teach any courses? And if you do, which ones would you like to mention?
So I do teach, although I'm in a research institute. That's because I would like to be connected with the university, and I would like to contribute also to the teaching, because I think it's important to teach the students what we know, and it's also interesting because you meet a lot of interesting people, a lot of students, and then some of them, they come back to your lab as a master's student, for example, which is good because you already know them and you've seen them in the classes. So what I do is I've created a new class or a new course in pharmacy, which is called Nanoparticles and Drug Delivery, which is basically an introduction to the field of nanotechnology for drug delivery. And then I also teach in a class for biotechnology and bioinformatics students, which is an introduction to biopharmacy. So it's comparable to the first one. It's also about targeted drug delivery, but it's more basic because they don't have the previous experience they're not pharmacy students, so you need to start further at the beginning. But still, both of them are interesting experiences, because turns out sometimes some of the students, they also mix, and then you have to adjust and you have to make sure that everyone in the class understands what you want to say, which was an interesting experience I had, I think, last semester or the semester before. And then I had to adjust my course, which I didn't expect before. So that was good.
Okay. So is it for like the chemistry students or physics students, like an introduction, or is it like
Yes.
for biochemical students?
It's more. So there's
Uh-huh.
another class that we organize irregularly, which is more advanced on vaccine development. And then if you want to take that class, it's advisable to also take the introductory class first to understand the general concept of what is drug delivery, why nanotechnology, why is it interesting to use this.
Okay. That must be interesting. So do you enjoy the teaching experience now?
Yes, I do, but it's always different. Every semester it's different. You think that sometimes, I mean, after a while you think you have some regularity or some more experience, but still then there's other people sitting there, and they still challenge you in a different way, which is very good, I think.
Yeah, it's like giving a conference talk on a regular basis, right?
Yes. Yeah.
Yeah.
Exactly. That's true.
All right. Okay, let's move on to your research experience. I hope your research experience so far has been wonderful and well, clearly because you're still doing science and will continue to be amazing in the future. Having said that, if you have three wishes to improve your research experience, what would you ask for? And I'm not promising anything here.
Yeah. um, So the first thing that I would like to say is that I really would want a experienced postdoc or a highly motivated postdoc to come into my group and to bring in some sort of new research concept or a different angle of looking at what we are doing. I've been trying this. It's not so simple for a junior research group. On one hand, you have to find the money, which is, I guess, the big challenge on one side, and then you also have to find a good person to come in and bring in new ideas. So if I could wish for that, that would also enhance the way that the entire group was doing research. I would also be interested in learning more on, let's say, molecular biology on extracellular vesicles. Not only look at them from drug delivery point of view, but they are also involved in physiological cell-cell communication, in development of cancer, for example, but also in transfer of resistance genes in antimicrobial treatment. And so we're not so experienced on the biology side, but I found it really interesting to better understand these concepts and to learn these methods. And the last one is not really research experience. It's more something that I also personally is looking for, is to find a permanent position at some point.
Okay, yeah, that makes sense. All three wishes of yours, they're very, very valid. And I wish I could just find a postdoc
Yeah.
for you that fits and just starts with you tomorrow or today.
Okay.
Or just do a magic wand and put all the knowledge of biology, extracellular vesicles in you. And lastly, and importantly, give you a permanent position, but I don't have that. But
Yes.
I hope and I wish you good luck with. I think these are all doable wishes. I think you are quite
Yeah.
realistic.
Okay. That's good.
I'm sure you'll make it happen at some point. Um, so speaking of future, what are you most looking forward to in the next 3 months?
So I think there's been especially one manuscript that has been sitting on my desk in the last year, which also deals with a very interesting topic and something that I think has not been reported in literature. There's been one or two master students that already worked on this, and they all have interesting results. But now it's my challenge to bring together all the results and then also write that paper and try to publish it in this time. And now I'm taking this time now because I'm in my home office, and I thought this is a good time to sit down and clear my mind and start writing this paper in a nice way. So that's what I'm hoping to do, to finish.
Okay, making the most out of the current situation, are we?
Yes.
Okay, all right. And before we let you go, Gregor, what we want to understand is what are the big challenges or what are the big questions that the researchers in your field are working towards solving, other than the vaccine for coronavirus,
Yes.
which is the situation at the moment. Other than that, what are the big challenges that we are working towards?
So I think what nanomedicine offers is the opportunity to personalize the therapy. If you think about cancer, for example, not every cancer patient is the same. Also, the same applies for bacterial infections. Not everybody reacts the same way to certain drugs. Some patients respond very well to the drugs, others don't. So what we can do, of course, is we can stratify the treatment in a sense that we are classifying the patients into certain groups, saying people that are responding well to the drug, people that are not responding well to the drug. But then I think if we develop smart or novel carriers, or we basically have the knowledge to do this, novel drug carriers, to better individualize the treatment, and to optimize and engineer our opportunities to take advantage of this what the nanotechnology is offering us, then I think we can really strongly enhance the way how different patients are treated, even if they have the same disease or if they have the same type of cancer or infection. So I think personalization is a major strength of nanomedicine that we can achieve.
Okay. It sounds almost very science fiction-y to me, that you can personalize the treatment for every patient. That would be so cool.
Yeah, I mean, I guess it starts with not giving everyone the same dose as it's done now mostly in the clinical treatment, because we know that women and men are reacting differently to things, and older people have metabolism, and so we can not only do this for the drug itself, we can also develop carriers that deliver the information based on what the physiological condition of the patient is, to make sure that in some patient this type of carrier will work better than in other patients, and we need to find out. But if we have the knowledge, we can better attribute the treatment, and we can also reduce the side effects of the treatments, which in most cases is already a lot. If we relieve the patients, for example, in cancer treatment, if you make sure that they don't have as much side effects, then this will help a lot the patient itself. And also it will be beneficial for the treatment.
Okay, that's we. I hope we are working towards this as fast as we can, and I hope it becomes reality soon. Thank you very much, Gregor, for speaking with us, and we are looking forward to having you on Real Scientist Nano.
Okay, thank you very much. Thanks for having me.
Thank you for listening. To know more about us, please visit our website realscientistsnano.org and follow us on Twitter at realsci_nano.